Judy asked me, why on earth did we pop out of the blue and invite you here? And the answer, Judy, is I couldn't resist romantic anatomist.
That was after.
Romantic anatomist.
Okay, I think we might need the lights down a little bit. My dad says I started to ask big questions a long, long time ago. One day walking down the street in the evening and noticing that the moon moved, but the stars didn't. And my dad just happens to be pretty good in physics and astronomy and math, and he gave me a lesson in parallax, and he said I was 2. And it took me until 1979 to see my first solar eclipse. That's a place where you see parallax in action, where the moon is moving across a star, and you see one of the most beautiful things in the world and out of the world. And why is a cell biologist starting with a solar eclipse? This is because I chase solar eclipses for a hobby, and I find them so inspiring because they make me think about what I might look like down here if I can look up there. And I always play a game with myself on airplanes. I'm flying over, I try to figure out where I am, and I try to think, if I didn't know where I was, but had arrived here from somewhere else, what I would think was going on, if anything, from up there, by the patterns that I see on the ground or in the sky with clouds and an eclipse. So that's a long way of saying that thinking is lateral for a scientist, as for everybody, I believe, if you're creatively motivated. I'm going to try to bring you to the point where you understand that coming to a hypothesis and having an idea, making a discovery, which, I've never made a discovery before. What the hell is that? The newspaper called me a scientist, I do a job. I teach, I create something, some kind of knowledge at a university. But my mom was thrilled by the newspaper saying I was a scientist. That was the best part. So what do? I teach anatomy in a medical school. I wanted to look at the clock because I hate being overtime. Yeah, okay. It's 1:45. So I teach anatomy, which means I go into the morgue and I teach classes dissecting people who have the foresight to give their bodies so that medical students can learn about the body, because there is only one way. It's a little gruesome. You saw some pictures yesterday in a different context entirely. Being in the morgue is a fabulous privilege. I get to learn over and over again how parts make a whole, and then think about those pieces allowing the whole to function as a whole. And I get to sit and stand and talk and dissect with the students and feel their thrill at learning about how parts make a whole and what structure and function can do when they work together. To me, that is an incredible privilege of being at a university. Another thing I teach is something called problem solving. In the medical school, that is a pretty good talent. I also try to teach this to graduate students who are the future scientists in this world. And it's something that, maybe you're thinking about being a graduate student in science or in anatomy, which I teach. It's something you pay a lot of tax for, but it's worth every dime, because you don't get anywhere without being able to think, and universities are a good way to do that. This is how I teach medical students and graduate students how to think. I have a class exercise where the beginning of the year I give out to all the students a bag of chocolate chips and a recipe. And we hypothesize, or we think about what should be happening. We make a hypothesis, or the students do, that we've got the same recipe, we've got the same chocolate chips, everybody's got an oven and butter and flour. Go make the cookies. The hypothesis is they are all the same. The next week, they have to bring a dozen cookies. We put them all on the table, and we figure out how we're going to judge what we like, what we do, what they look like, what they taste like. And these cookies are all from the same recipe. This one on the bottom, the kid ate the chocolate chips, substituted. He substituted chocolate ice cream because it was chocolate. He didn't know what the hell he was going to make, but they didn't score so well in the ranking. From this, we then think about why they're different, and we make more hypotheses. So we get a hypothesis, that frames the questions, then we get the data, then we figure out what went wrong, and we get another question that we can test in a different way. This isn't rocket science, but it's science. We published this, actually, and I presented it at a conference by handing out cookies and asking people to tell me which one they liked best and why. But I think that there are really important clues in everything we do. Everything we have around us is something we can ask a question about. And I never understand why people don't ask questions. Nope, wrong button, wrong place. This is a muscle fiber, a single muscle fiber from the toe of a mouse with muscular dystrophy. And it's not so that I can tell you all about what's happening in here. This is one cell. Muscle fibers go from tendon to tendon. If you've got really long legs, you've got really long muscle fibers. They're essentially one cell with a lot of nuclei. This happens to be a cell from a mouse that has muscular dystrophy. And muscular dystrophy, for people who don't know, is a lethal genetic disease. It kills young men like war in their early 20s. And those children know from 3 or 2, or if they're in a certain part of the world from when they're born, that they have muscular dystrophy and that they will die. At least that's what happens to them now. And my goal in muscle research, because I study muscle research and muscle repair in mice, is that what I do in mice might help people with muscular dystrophy who will die. And many people are working on this, not just me. Please don't get that impression. I'm like a little tiny, tiny part of this. How does muscle start? Muscle starts by being a whole bunch of little cells that divide a lot when you're inside mum. And they all kind of line up and get together and make long lines, and somehow or other they know the axis to grow on, and they line up and they fuse together into a muscle fiber. And then they attach themselves to the bones that have to move. Muscle makes you move. Without muscle, you don't move, you don't talk, you don't breathe, you don't shit, you don't make love, you don't look at solar eclipses. Muscle is pretty important. It's heart muscle, too. Better not forget that. None of those nuclei, because a muscle has many nuclei inside it, because they all fuse together, none of those nuclei can divide. They keep house. They make your muscle contract. Muscle shortens, and then it relaxes and it can lengthen. But they can't divide. What saves us if we injure muscle, if we have surgery, or if we hit something, is a thing called a satellite cell. And I really like that it's called a satellite cell because, like, the moon is a satellite, and we all have satellites, and being on the stage actually is like having satellites all over. Satellite cells are the only cell in muscle tissue, in skeletal muscle, that can divide. So they have to turn on in order for you to get new muscle. And we've been studying muscle repair, what happens, like a day after injury, or 2 days after injury, or 4 days after injury, because every one of those little steps kinds of goes back to making new muscle. We make more little cells, we fuse them together in the right axis, and we put them onto a bone, and then they can work again. And at the end of a talk on muscle repair, because that's what I've been doing for 15 years, is learning about that, somebody in that audience who was really listening and really could think would say to me, "What turns that on?" And I would say, "You know, we don't know." And that was the dogma that I came into the muscle field with. That's what I decided to tilt against, my windmill. So what turns on satellite cells? Now, I had been studying these satellite cells here in green, which is sort of drape over a muscle fiber, which is black. Satellite cells have a receptor called c-Met, which means nothing to you, but it here is stained with an antibody to that protein. And the thing which is going to bind that receptor, wouldn't you know, it just happens to be this red stuff, which is all around the outside of muscle fibers, and that's called hepatocyte growth factor. Why it's called hepatocyte? It doesn't really matter what it's called. The first person who called it was studying liver, that's where it was. So to keep the nomenclature right, it's sort of hepatocyte growth factor. When hepatocyte growth factor binds the receptor, c-Met, which is in satellite cells, they scatter, they move. So I thought, because of some earlier work, that that movement thing was really important. And I guess that's what I was thinking about one day when I saw a poster for a talk. At universities, one of the neatest things is that you have the opportunity to see many, many people from all over the place and learn something. And you know, believe it or not, I go to lectures to learn a couple of things. I can't do 5, that's political. 2 is where I head. So I went to this lecture on liver repair. Now liver, you know, what on earth am I doing going to a liver repair talk? Well, this is hepatocyte growth factor, and that's from the liver, even though it's made by muscle. So I thought, well, I might learn something about liver and muscle. So I went to a talk, and I listened to liver repair, and I transposed it into muscle and activation because of what I heard there, which was totally amazing. I learned 2 things. I learned that nitric oxide is released from liver, muscle. I learned what I learned, so I'm going to say it muscle. It's released from muscle all the time, and there has been a number of people working on this over the years. Every time a muscle contracts, because of where the enzyme is located, just under the membrane, there, that black line, that's where the enzyme that makes nitric oxide is. And nitric oxide is a tiny little gas that just about every cell makes. But I learned that nitric oxide is released regularly. And one of the big things that releases nitric oxide in liver is shear. And shear is moving across. It actually feels pretty good in a lot of circumstances. But shear is lateral movement across each other in layers. And if ever there was a tissue set up for shear, it's muscle. And that's where I put together biophysics, as I understand it, and cell biology, and liver, and nitric oxide. And really, I didn't really do anything. I just took 2 neurons that were on the opposite sides of my cortex, and I put them up against each other, and they sparked, they talked. And I thought, well, shit, you know, if nitric oxide is released on shear, and you break a muscle fiber by cutting into it, then the 2 sides of it, actually, because all kinds of calcium rushes into the muscle fiber, the 2 sides will actually contract and pull, retract back. If you cut your leg muscle and it's alive, it will recede, and that wound will open really big. Anybody ever cuts himself, they'll now know why. This is shear. There's shear, there's a sleeve around this muscle fiber. It's really quite tight, actually. There's a membrane around the muscle fiber that holds all this stuff in that's all contracting and relaxing all the time. there's a satellite cell that normally sits in between them, and it's actually attached to both of those layers. So when the fiber contracts within the sleeve and the sleeve doesn't contract, the cell is like a pea rolling between those two layers. And that's a setup for shear. At least that's what I thought. So I thought, well, you know, this is a really outlandish thing to say that this is going to turn on satellite cells. Like, totally out of the blue. So I thought, why? I don't want to waste a lot of time on this. If it's so out of the blue that it shouldn't be in here, we'll get rid of it quick. We've got to figure out yes or no. So I designed what I now think of as a dumb assay. I wanted the cells to tell me yes or no. If it was yes, I would go for it. If it was no, I would dump it fast. Because wild goose chasing is too expensive in science. We don't get enough of those research grants to do basic research because it's so hard to portray what it means until you figure something out. So let me say that my assay was to figure out movement of cells. And I figured that if nitric oxide did something, that putting it in there would make it happen fast. And we know that cells have to be activated quite quickly in order to start muscle repair the best way they can. And I also figured that if you shut off nitric oxide production just before an injury, that you would not get that movement. So that was my test. You could turn it on, you could turn it off. Kind of like figuring out that you're working with the right circuit when you're repairing your bathroom and cutting the wires just to make sure, because your house is so old, there's no such thing as a wiring diagram in it. Okay. So we asked these mice. We set up the experiments. The technician was fabulous. The students were fabulous. We just designed this so it would be double blind, which means nobody knows except the mice. And they told us. They said, it's nitric oxide. Right away, within a minute of injuring these cells, you get shear, you get a huge lump of nitric oxide come out, and then silence. Nothing. And it's that whoosh, boom, transient that is the signal. And that happened within a minute, as fast as we could get the muscle out of the mouse, do something with it, and count how many cells came off, were moving. That's how fast it happened. And not only that, but if you add something in to make that happen, you get muscle growth. That's what CFRB in Toronto phoned me at 6:05 the morning that it came out on the National Post and asked me about Schwarzenegger mice. And, you know, I go to the gym like you go to the gym, not very often, not as often as I could, because I'm a really nerdy scientist, but you know, that's what a lot of people are interested in, muscle. That's what a lot of other people, including the space station, phone me about coming to the lab with their TV cameras, like, this is really off the wall for me in an anatomy department, which is pretty dead. To have a television camera to record life and mice, that's pretty exciting. Anyway, where is this going? An awful lot has happened since May the 1st, when that story broke. And I've got to tell you that because you can grow muscle this way, and because you grow muscle this way already, there's already an application. You are it. When you exercise a lot, you're using that mechanism because you're making muscles shear more. I think when we add this stuff in, so there's more nitric oxide in muscle with muscular dystrophy, at least our mice tell us this is true, we can actually make those muscle fibers repair better and reduce the amount of muscular dystrophy that those mice have at the age when it should be getting worse fast. And this has been just a huge insight into what on earth I've been doing for 15 years, thinking about muscle repair and why it was happening the way it was happening. Just what? Jeffrey Simpson wrote this in connection with a bridge, but all the time people are using analogies to muscle and muscle flexing. They really are getting at the heart of a matter. There is a huge leap between a discovery and a treatment. And I got 700 emails since May the 1st asking me about this, from people whose kids have muscular dystrophy, from people who are dying of ALS, from people who have MS, from people who have had strokes, from people who want to get into the RCMP so they need bigger muscles. From everybody, from Turkey, from Australia. Just blew me away. There's too much water under the bridge between politics and universities and education and basic research. And you and I have to work on that together. So we've got to flex some muscle. We have to make this work. I mean, I don't want to have a vested interest in this, don't get me wrong. I put a patent on this because the only way anybody will benefit from it is if there's a way for somebody to license it and make money. And as a scientist, that interferes. But I realize now that the paradigm is way out there. That has to be the way we're going. So part of this is a plea for you to be involved in getting active with politicians, with educators, with universities, and with the scientists in your community, because they can all use your encouragement. This has been incredibly encouraging to have that much attention and even not want it after a while, because it's a little hard to deal with when you really have to do some more experiments. So there's water under that bridge and we have to work on it. But I want to leave you with the idea that having made a discovery like this, totally out of the blue, and as someone said yesterday, thinking out of the box, has just been a revolution in me. And that now I see exactly where I want to go with science. I see hypotheses just straight back, like looking at those mirrors we saw yesterday at the Children's Holocaust Museum. I can see so many lines, and I know what I have to do for the next 30 years if I should be so lucky. I know that I have to do that. And if I can take my inspiration every year, because there's about one of these a year, from looking up and from looking through a microscope down at tiny little things, I think we have to look both ways all the time. So why am I here, Moses? The reason I came up with this is I'm here to ask you to look both ways and to activate your satellites, your students. We all have students. And to keep dreaming. Thank you.